Okay, so welcome to this next video in which we are discussing the VIP and PACAP receptors. Okay, right, so we've seen now that when VIP and PACAP bind to these VIP and PACAP receptors, what happens is the PACAP and VIP receptors interact with heterotrimeric G proteins of the GS form, and this leads to the activation of adenyl cyclase enzymes and the production of cyclic AMP. Okay, the cyclic AMP is now going to go and activate a very famous enzyme known as protein kinase A. Okay, so the next topic is indeed protein kinase A. Right, so protein kinase A is not just one protein. Instead, it is what is known as a hollow enzyme. It's made up of multiple proteins stuck together, at least in the inactive form. Okay, so I want to start by showing you the inactive protein kinase A hollow enzyme. And then we'll discuss the activation of the protein kinase A hollow enzyme, and then what's going to actually happen once you have activated the protein kinase A hollow enzyme, i.e. what does it then do? Okay, so the protein kinase A hollow enzyme is made up of 4 separate proteins. Okay, it's made up of 2 subunits that are known as regulatory subunits, which form a dimer, and then you attach to this dimer of regulatory subunits 2 catalytic subunits. Okay, so let me draw pictures of this. So we'll start off by drawing a single regulatory subunit. We'll then show how they dimerize together, and finally we'll show how the catalytic subunits join the fun, basically. Okay, right, so here's the amino terminus of a regulatory subunit polypeptide. The first important domain that you then have at the amino side of the polypeptide, which I will colour in turquoise here, is known as the docking and dimerization domain, and I'm just wondering where I want to draw this. Okay, I'll put this label here. So this turquoise domain is called the docking and dimerization domain. Okay, and this is going to be important in dimerizing the 2 regulatory subunits together. It's also going to be important in docking the regulatory subunit dimer to proteins known as A kinase anchoring proteins, PACAPs, which we'll come to later. Okay, so wait and see what this domain does, and I'll show you. I hope it'll be clear soon. So the docking and dimerization domain is also called the D/D domain. Okay, D for docking, D for dimerization. Okay, so the docking/dimerization domain. Right. Then the next bit that you have is what's known as a protein kinase A inhibitor site. Okay, and this is the portion that's involved in binding to the catalytic subunits of the protein kinase A hollow enzyme, and which will inactivate them in the inactive protein kinase A hollow enzymes. Okay, so this is known as the protein kinase A inhibitor site. Okay, so each regulatory subunit will have a protein kinase A inhibitor site. Then the 2 next domains which I wish to talk about are the cyclic AMP binding domains. Now you have 2 of these and they're next to each other, and the fancy way of saying that you've got 2 next to each other is to say that they're in tandem. Okay, so these are the 2 in tandem cyclic AMP binding domains, and I'll try and squash this in down here. Okay, so these are binding domains for cyclic AMP. So this is how cyclic AMP is going to interact with the protein kinase A hollow enzyme to cause a change. Okay, right. And then after the 2 cyclic AMP binding domains, you then have the carboxylic acid terminus of the regulatory subunit, and then that's the regulatory subunit finished. So this, what I've drawn you now, is what's known as a regulatory subunit of protein kinase A, and I really should have started a new piece of paper. Regulatory subunit of protein kinase A. So it is 1/4 of the protein kinase A hollow enzyme. The whole thing is going to be made up of 2 of these and 2 other subunits that we haven't even discussed yet. So what's going to happen is you're going to take another regulatory subunit of protein kinase A, and they're going to dimerize together. Okay, so here is the other one that I'm putting in here now. Okay, and the way they'll bind together is the two docking and dimerization domains will bind like so. Okay, then the rest of the polypeptide will go off down here. So again, here is another protein kinase A inhibitor site here. Okay, and then after the protein kinase A inhibitor site, the regulatory subunit here will also have two cyclic AMP binding domains which are in tandem, like so. Okay, and then they'll have the carboxylic acid terminus right at the end down here. Okay, and I'll colour these cyclic AMP binding sites in blue again as well. Okay, so each one has two binding sites for cyclic AMP, which means that two cyclic AMP molecules can bind to each of the regulatory subunits. Okay, so what we've created so far is called a regulatory subunit dimer. Okay, so we haven't finished the whole protein kinase A holoenzyme so far, but we have got a regulatory subunit dimer. Now, before I go any further, what I want to tell you is about the different types of regulatory subunits. Okay, so there is not just one regulatory subunit of protein kinase A. It would be beautifully simple if there was just one regulatory subunit of protein kinase A, but it's not the case. There are multiple regulatory subunits of protein kinase A. So there are 4 in total, and they're grouped into 2 major families. So there are the type 1 regulatory subunits of protein kinase A, known as the R1s, for R for regulatory, 1 for 1. So these are the type 1 regulatory subunits. And the second family is known as the type 2 regulatory subunits. So what are the members of these 2 families? Well, in the type 1 regulatory subunit family, you have the type 1 regulatory subunit alpha, and then you also have the type 1 regulatory subunit beta. And in the type 2 regulatory subunit family, you have the type 2 regulatory subunit alpha, and then also the type 2 regulatory subunit beta. Okay, right. So, the question then arises, how do we build these regulatory subunit dimers? Because it was very simple when we were under the misbelief that there was just one regulatory subunit of protein kinase A. All you had to do was then take 2 copies of this and dimerize them together, but now we've got 4 types. That opens up a whole new wave of possibilities, because do you have to have the same regulatory subunit here as here? Well, the answer is no, not always. Okay, so basically, you can form homodimers. Okay, so if you have 2 identical regulatory subunits in the 2 slots here, so this one is identical to this one, then it's called a homodimer. Homo means same, dimer means 2-membered structure. So this is a 2-membered structure, a dimer, where both of the regulatory subunits are the same. Okay, right, and you can form homodimers for all 4 of these. So there is a type 1 regulatory subunit alpha homodimer, there's a type 1 regulatory subunit beta homodimer, there's a type 2 regulatory subunit alpha homodimer, and there is a type 2 regulatory subunit beta homodimer. Okay, then there is one type of heterodimer you can form. Okay, so basically, you can form a heterodimer where you join type 1 regulatory subunit alpha to a type 1 regulatory subunit beta. Okay, so you can also make regulatory subunit dimers where you have one type 1 regulatory subunit alpha here and another type 1 regulatory subunit beta here. So you can form that one heterodimer. That's the only heterodimer you can form, though. So most of them form homodimers. Okay, right. Now, we will talk about what the significance of the different family of of regulatory subunits is in a moment. But know for now that if you build a protein kinase A holoenzyme which has type 1 regulatory subunits. Okay, so it's either a homodimer of type 1 regulatory subunit alphas or a homodimer of type 1 regulatory subunit betas, or this one heterodimer where you have a type 1 regulatory subunit alpha with a type 1 regulatory subunit beta, then that's known as a type 1 protein kinase A holoenzyme. Okay? And if you use type 2 regulatory subunits to make the regulatory subunit dimer that's going to be the backbone for your protein kinase A holoenzyme, then the overall protein kinase A holoenzyme that you end up with is known as a type 2 protein kinase A holoenzyme. Okay, right. Now let's discuss the catalytic subunits then. So this isn't the complete protein kinase A holoenzyme yet. We've just got the regulatory subunit dimer. To make it into the complete protein kinase A holoenzyme, you need 2 additional subunits to bind, and they're both going to bind to the protein kinase A inhibitor sites of the regulatory subunits, and these are called the catalytic subunits. So let's colour these in orange here. Okay, so you have 2 catalytic subunits that then come and bind to the protein kinase A inhibitor sites of the 2 regulatory subunits that are dimerized together within the regulatory subunit dimer. Okay, there are then 3 different types of catalytic subunits of protein kinase A. There is the catalytic subunit alpha, there is the catalytic subunit beta, and then there is the catalytic subunit gamma. Okay, so in each of these slots you have 3 different catalytic subunits that you can use. Okay, so this is quite complex. If you want to build a protein kinase A holoenzyme then, you have to firstly build your regulatory subunit dimer. Okay, you firstly decide, do you want to go down the type 1 route or the type 2 route? If you go down the type 1 route, there are many options available for your regulatory subunit dimer. You can either make a homodimer, either of type 1 regulatory subunit alphas or of type 1 regulatory subunit betas, or you can make a heterodimer which contains one of each. Okay, if you go down the type 2 route, there's less options available for you. You can only make a homodimer. So you can make a homodimer of type 2 regulatory subunit alphas or type 2 regulatory subunit betas. Then you have to add on your 2 catalytic subunits, and for each of these you have 3 options available. You can either put in a catalytic subunit alpha, a catalytic subunit beta, or a catalytic subunit gamma. So these are separate proteins in their own rights as well. And these are the portions which are actually going to catalyze the addition of phosphate groups onto serine and threonine residues within proteins. Okay, right. So before we discuss the activation of these protein kinase A holoenzymes, for at the moment they are completely inactive. Once the catalytic subunits are bound to these protein kinase A inhibitor sites, they do not do their job. They do not put phosphate groups onto serine and threonine residues within proteins, so they're not active at the moment. They're held inactive by the regulatory subunits. Okay, we'll discuss the activation in a moment, but firstly I want to discuss the significance of this distinction between type 1 protein kinase A holoenzymes, in which the regulatory subunit dimer is made out of type 1 regulatory subunits, and then also type 2 protein kinase A holoenzymes, in which the regulatory subunits are made out of type 2 regulatory subunits. Okay, right. So basically, type 1 protein kinase A enzymes are generally free within the cytoplasm. So this holoenzyme that I've drawn here, if these regulatory subunits in the regulatory subunit dimer are made out of type 1 regulatory subunits, okay, then the holoenzyme generally is free within the cytoplasm. Whereas if you have a type 2 protein kinase A holoenzyme, which means that the regulatory subunits which make up the regulatory subunit dimer are type 2 regulatory subunits, then these are generally bound to the underside of membranes, basically, mainly the cell membrane, but they're also attached to other membranes, such as the membranes of the intracellular organelles. Okay, now the reason for this is that type 2 regulatory subunits, the docking and dimerization domains of type 2 regulatory subunits bind to proteins known as PACAPs. Okay, and I'll come on to this on another piece of paper. Okay, right. So, attached to membranes within cells, and I'll use as an example the cell membrane, because that's the main example, but intracellular organelle membranes also have these. Okay, so attached to the underside of the cell membrane, okay, and facing into the cytoplasm, you have proteins that are collectively known as ACAPs. Now, by no means is there only one ACAP. There are a huge number of different ACAPs that have been characterized, okay? But for our purposes, we'll just keep them in full generality, okay? And ACAP stands for A kinase, referring to protein kinase A. And then the second A stands for anchoring, and then the P for protein. So the ACAPs are the A kinase anchoring proteins. And basically, these combined to the dimerized docking and dimerization domains of a protein kinase A holoenzyme of the type 2 form. Okay, so I'll draw this here. So here is the amino terminus of our regulatory subunit. Okay, here is the docking and dimerization domain, which will be dimerized with another docking and dimerization domain of another regulatory subunit. Okay, and then it will continue on. We'll have the protein kinase A inhibitor sites here. So let's color this in. Okay, so in purple here are the protein kinase A inhibitor sites. In turquoise here are the docking and dimerization domains. In blue here is our ACAP, okay, which was a bad choice of color because I colored the cyclic AMP binding domains in blue as well, but never mind. Okay, so here are our two cyclic AMP binding domains that are in tandem, okay, like so, and then here is the carboxylic acid terminus of this regulatory subunit, and then we'll have the same on the other side. So here's our first cyclic AMP binding domain, here's our second cyclic AMP binding domain, here's our carboxylic acid terminus. Okay, I'm going to colour that in blue again. And then, of course, this is just the regulatory subunit dimer. We need now to add on the catalytic subunits to make our full protein kinase A holoenzyme. Okay, so here then are the catalytic subunits bound to the protein kinase A inhibitor sites of the regulatory subunits of protein kinase A. Okay, so here are the catalytic subunits highlighted now in orange. Right, okay, so here is our protein kinase A holoenzyme. Now, if it's a type 1 protein kinase A holoenzyme, i.e. if the regulatory subunits are type 1 regulatory subunits, then it will not do this, or at least it hardly does this. Okay, it's not true to say that it doesn't do it at all, but the affinity for binding to ACAPs is ridiculously small for type 1 protein kinase A's. So, to a decent enough approximation, type 1 protein kinase A's are free within the cytoplasm. Type 2 protein kinase A's, however, where the regulatory subunits are type 2 regulatory subunits, the dimerized docking and dimerization domains here are going to now bind to the A kinase anchoring protein. So this will bind to this dimerized set of docking and dimerization domains, and it will hold the whole protein kinase A holoenzyme at the membrane, basically. Okay, which in this case is just underneath the cell membrane. Okay, right, so that's the function of A kinase anchoring proteins, and that's the significance of the difference between type 1 protein kinase A holoenzymes and the type 2 protein kinase A holoenzymes. Okay, so in the next video what we'll do is we'll turn our attention to the activation of of protein kinase A holoenzymes, both type 1 and type 2, and then what they actually do once they have been activated.